Good morning, student. Our topic is today introduction of recombinant DNA into host cell. So there are different techniques are present for introduction the recombinant DNA inside the host cell. Based on the host cell type, we generally use two different terminology. If it is a bacterial cell, we generally use the word transformation. If it is the animal cell culture, then we are using the terminology transfection. So there are different like chemical delivery method, physical delivery methods are there. We will start with some of the important topic in the chemical delivery method. The most popular one is calcium phosphate mediated transfection. Transfection meaning we are dealing with the animal cell line. So what we do generally, we incubate the DNA in phosphate buffer. We may take some of the other unrelated DNA like salmon sperm DNA in order to increase the volume, in order to reduce some non-specific interaction, and then incubate with calcium chloride solution. Mix these two things gently, and then precipitate this one drop wise. We are putting in the our cell lines on our cell line, and we need to incubate for 48 hours in an incubator, and then we need to check the cells for the selection marker whether these cells are being transfected or not. Like calcium phosphate transfection method, there is one more, is that polyplex, like that DEAE is a dextran which actually used for ion exchange chromatography, is a positively charged column resin. So you can see the negatively charged DNA can easily bind with that, and this name is diethyl aminoethyl dextran. So what we do in presence of this kind of positively charged resin, and when it is bounded with a negatively charged our recombinant DNA, it actually induces an endocytosis inside the host cell. Presence of this thing will cause an endocytosis inside the host cell, and the host will take the foreign DNA molecule inside the cell. There are another method which is very popular method, that is cationic liposome, LipPerfectin, lipofectamine, lipofectase, DDAB. There are many commercially available lipofectin. This is a lipid bilayer micelle kind of thing, which is form of a micelle kind of thing, where inside our gene of interest or our clone DNA is there, which also induce an endocytosis inside the cell, and can take this micelle can get fused easily with the lipid bilayer, and our gene of interest can go inside the host cell. So our next topic is physical delivery method. There are many physical delivery method. One is electroporation, microinjection, gene gun, that is microblastics or biolistics method. So what is the how the efficiency will increase by using this physical delivery method? You can see that here are letters. Earlier what we are doing, we are incubating the DNA and waiting for the endocytosis to take place. Instead of doing that thing directly, we can introduce with the help of some physical delivery method inside the cell. This device is actually used as a gene gun. The most popular method for this is electroporation, microinjection. Now electroporation primarily used for cells in suspension, that is hematopoietic cell lines and embryonic stem cells, and has also been successfully used for transfection of fibroblast and airway epithelial cells. So different types of cell line people have used and treated for electroporation. I will come to the point later in detail. And then microinjection, generally oocyte or zygote transfection, effective for embryonic stem cells and airway epithelial cells, being used for microinjection. People have done plant cells also for microinjection. Now little details about electroporation. You can see the method. You need to give electric impulse voltage certain amount of, so that there will be a temporary hole formation in the cell membrane during that pulse, and it is a temporary transient thing. So once you will withdraw that electric pulse, hopefully the cell membrane will be again repaired, and by that time, because of these holes are formed, the gene of your interest or the vector recombinant DNA will go inside the cell. So it's a controlled millisecond electrical pulse induce temporary pores in the cell membrane, and cell membrane reseal, and it's left unharmed. Most of the cases it's unharmed. This is the machinery where we are doing. Aseptically, you need to take your culture and you need to incubate with your recombinant DNA, and you need to give a pulse based on type of cell, and this is the mechanism principle behind the electroporation. Another very popular chemical method which we regularly use for our transformation, plasmid, our recombinant plasmid to introduce inside the bacterial cell, that's named as chemical transformation with the calcium chloride method. So this is bacterial cell again, transformation I'm talking. So bacterial cells generally they don't like to take any foreign DNA, but in case it is taking a foreign DNA, you need to make the cell competent enough to take a foreign DNA. So there is a procedure. Treating with ice cold calcium chloride solution will make the cell, bacterial cell I'm talking about, equally competent. That means this divalent ion temporarily make some this in the very cold condition, temporarily make bind with the plasma membrane and make some hole, and the hole not open, but we need to give a heat shock at 42 degree centigrade. Then this heat shock will cause the pore to open, and that time if you incubate with your donor, that means the vector, the recombinant DNA, that will go inside, and you need to remove the and then you are going to plate it. So little bit details if I go, you can take a log phase E. coli culture, healthy cell you need to take, that's why log phase. You centrifuge it, you need to take the suspension of the cell and treat it with ice cold calcium chloride solution. Please mention ice cold condition. Now this will treating with ice cold calcium chloride will make the cell competent cell. Then you can aliquot different cell and you can store it at minus 80 degrees centigrade. Now you take one vial from out of this and now introduce your incubate with your ampicillin resistance plasmid DNA, that is the recombinant DNA. Now after that, once the cells coming, I mean you are taking from minus 80 degree, you are need to thaw it in the ice. So after that you need to give a 42 degree heat shock. So that's in presence of calcium, and this heat shock the pore will be open, and we are the mechanism behind is that during that time only this vector will get introduced inside the cell, and based on the selection marker present here, ampicillin here present, so you can plate in the ampicillin containing plate. If the plasmid has gone inside, then only the bacteria will grow in the ampicillin containing plate. So you can screen it also. This is a very common technique. You can do blue-white screening also if time permits, if everything goes right. So we will do this experiment in laboratory. Now this is another way. This is the chemical method we have described. Now this is another, just what we described, the electroporation. So same way you will be taking the log phase cell, centrifuge, suspend it. You are taking in a aliquot in a bigger amount, not a small amount, because I need lots of cell in order to do electroporation, because when we are applying the voltage, many cells will die. So what we are doing, we are taking the cell and giving in an incubator. The incubator is having actually a cuvette. You have seen the cuvette for spectrophotometric when you are doing spectrophotometric experiment. So that type of cuvette you are taking. In the cuvette there are two electrodes are already there, that can be connected, and you can give the impulse, and then you have to select in a proper selection media. Our next topic is microinjection. This is a technique where a very low copy number vectors are directly introduced inside the cell nucleus with a very fine needle, and this is the outline where the cell you have taken, you are seeing through a microscope, and directly injecting inside the nucleus with the needle. But actually this instrument looks like this. It has a robotic arm. You are only guiding the cell where to inject, and the robotic arm will do the rest of the function for you. Our next topic is bioelectric microprojectile gun. This is also named as gene gun. Little bit about this bioelectric particle delivery system. It is a device used to deliver exogenous DNA. Generally, DNA are coated with gold particle or sometimes tungsten gene. You can use DNA, you can use RNA or protein also to cells. By coating particles of a heavy metal with the gene of interest, and then you are firing this micro projectile into cell using mechanical force. So what is happening, you are your gene of interest is coated with a heavy metal, sometimes gold. People have seen that they are using tungsten gene also. So you need to fire it. So you can see in the diagram there's a firing mechanism. This is a vacuum where helium gas they have introduced, and with the help of helium gas only, I will tell you a little bit more details. Helium fills this chamber. Which chamber? You can see this barrel, this inside helium gas, this filled with this chamber. The pressure eventually reaches to the point where the rupture disc, there is a rupture disc towards the ruptured disc, and then the result, because of this pressure, it burst the helium, this DNA coated, gold coated particles into the stopping screen. Now what will happen, the bullet with what you are firing, that bullet will stop in the stopping screen, and all the DNA particle which is coated with gold particles will come to the towards the plant tissue culture where you kept your petri plate, and that will be inserted inside the cell, and the gene will be introduced inside the cell by using this technique. Our one more technique I'm going to discuss. This is we have already studied. This is called liposome mediated LipPerfection, also we can say. We are showing here the micelles where inside phospholipid bilayer, and inside our gene of interest, actually our recombinant DNA, we are incubating, and this is the cell line, fibroblast cell line they have taken, DNA being negatively charged, and these are the cationic lipids. If you incubate, the DNA will go inside, and that will be ultimately endocytosis and go inside the host cell. So if your recombinant DNA is carrying one particular marker, next step is to screen the cells which have taken your gene of interest. So with that, these are the techniques we have discussed. Our next topic will be how to form a genomic DNA library. So I hope you are studying, and any confusion, any doubt you have, please ping me, and you can call me also. I will definitely try my level best to answer you. Thank you very much.